All right, here we go with the final chapter, the final installment for chapter 6. So 6.3 focuses on apoptosis. And just to get that out of the way straight away, the pronunciation of this word is apparently a very contentious issue in the world of biology. I am an apoptosis girl. Because it is much more fun to say than apoptosis or apoptosis. If you already have a way that you say it, I'm sure anything is fine. But I will firmly remain an apoptosis person. I'd love to know what you are, Miss Markham. And it's just in the last few days that this whole big online argument about how you pronounce apoptosis has come up in the world of biology teaching and biology students. So decide which side of the fence you sit on. Let's have a look at what the process actually is. So just move me slightly there. Okay, so apoptosis is what we call regulated or programmed cell death. So it's a process by which it's very orderly. It's where a cell is either required to die after some damage or has always been programmed to die where it needs to be replaced, or it's just no longer useful. So programmed cell death. Why do cells undergo apoptosis? Lots and lots of reasons. So during growth and development, there's so many tissues that need to be turned over or are no longer required. So tissues of the umbilical cord or tissues in the webbing in between fingers as the embryo is developing. They are cells that are eventually no longer needed. So they are programmed when they are developing. They have it written into them that this is going to result in cell death, this growth and development. It may be something that is happening throughout the course of your life. So skin cells, blood cells, things that need to be replenished and replaced over time, they will undergo apoptosis. And it could be cells that have incurred some damage. So that could be damage from sunlight even. And then there are pathways and triggers that are set off that will initiate apoptosis. So the main part of what you need to gauge about apoptosis is one, the steps that a cell undergoes to follow this pathway of apoptosis. What is this regulated pathway and what does it look like? What does it involve? Appreciating that the signal for this this process can come from inside the cell or outside the cell. That is a really big idea to get the difference between. And it is being used as an example that you can apply that signal transduction response model to. So being able to talk about apoptosis in those 3 steps that we're getting used to, that's an important idea as well. And the last one that I've got listed there is being able to contrast it with necrosis. So if it's not a programmed, regulated, nice, orderly cell death, the other option is this necrosis, which is a violent, very contradictory to apoptosis kind of death for a cell. So let's have a look here. Okay. So being able to discuss apoptosis was a good spot for me down here. Being able to discuss apoptosis in that model of reception, transduction, response, that's something that you want to put into practice and be able to do off the top of your head. I just kind of want to skip forward to the next slide first because we get a nice picture of apoptosis. So here we've got apoptosis in diagram form. So we go from a cell that is healthy to a cell that has begun this process of blebbing. And that's a key term to use to describe that misshapen kind of structure that our cell begins to take on. which progresses further into the cell actually breaking apart into what we call apoptotic bodies. And in those apoptotic bodies, you'll find the organelles, the broken down pieces of that cell, nice and neat in these little packages. And then those packages, those apoptotic bodies, will eventually be cleaned up by something that we're going to become very familiar with, a phagocyte. So the only place that you would have heard this term before in this course is probably when we've been talking about endo and exocytosis. So I did mention that if we're talking about endocytosis, so engulfing material, there's two kinds of endocytosis. It's called penocytosis if the material being engulfed is a liquid. It's called phagocytosis if the material being engulfed is a solid. So this is a perfect example. These are classes of cells whose job it is to engulf solid material. So these things called phagocytes, and there's a number of them that we're going to get to know in the immune system, but just for now we can generally refer to them as phagocytes. And I just had to quickly go and find this video that I found the other night while I was putting our new style together for this week. Just showing this real life picture of what a cell looks like as it's undergoing apoptosis. You can really nicely see here the cell beginning to form those blebs, which are breaking away from each other, all these blebs forming on the side, and they'll start to break away from each other and form those apoptotic bodies that will then be engulfed by the phagocyte coming through. So just to put into a real life sort of context what the cell looks like as it's undergoing apoptosis, you can see those apoptotic bodies starting to break away on the side of the cell there. So you get and I'm a bit all over the place with this video, but I did forget at the start to go back and have a look at the key knowledge. So before I go any further, I'll just skip back a few slides and have a look again at the key knowledge that we need for this chapter part. All right, so we're going to get to know apoptosis as a natural regulatory process of programmed cell death, as I've already discussed, initiated after a cell receives a signal from inside or outside, and we're getting to that part, which results in the removal of cells that are no longer needed or may be a threat to the organism. It is mediated by enzymes, caspases. And the part that I really wanted to get to is where it talks about after this. So caspases is something we do need to know about. And what caspases do is they cleave specific proteins. I'm going to mention that again in a minute. In the cytoplasm or nucleus. Details of specific cytoplasmic or nuclear proteins are not required. So again, it's got that nice little detail in brackets there, telling us that you don't need to commit to memory all of the other enzymes that are involved in the process of apoptosis, but you do need to know about this one called caspases. So that's nice to know at the outset, what you're actually needing to pay close attention to. And then the only other dot point related to 6.3 is that malfunctions of apoptosis that result in deviant cell behavior, leading to diseases including cancer. So there's three sort of malfunctions that we'll talk about related to apoptosis after we've gone through the process. Just had a thought. Yeah, it is too. The signaling example that I used right at the start, actually forgot that I'd used an apoptosis example. So going back to that and looking at how complicated this process really is, we're refining it back to the really simple form. And where you see these CASP abbreviations here, they're those proteins that it was referring to, that the caspases, and you can see how many different parts of apoptosis they're involved in. So generally knowing what a caspase does and that they are very important in the signaling pathway for apoptosis. But that's not something that you need to stress about. That is the very complicated version of apoptosis, not what we need. And just before I go any further, I'm going to have a look at your textbook to get a little bit of an appreciation of that difference between apoptosis and necrosis. So where we see necrosis discussed here, you can see that comparison between apoptosis and necrosis. So where we're getting that nice packaged product of apoptotic bodies, which can be gotten rid of very quickly and easily by our phagocytes. Necrosis, in contrast, the cell actually ruptures. So any toxic material that's being contained in that cell has now gone and covered the nearby cells as well. And so what results is a lot of inflammation of the area. So that's a very nasty example of necrosis that it uses there, but that is the comparison. So apoptosis allows a cell to be gotten rid of quickly and neatly, whereas necrosis can result in really catastrophic infections inside someone. So that's that little comparison between apoptosis and necrosis. Okay, so we've kind of got an idea of the stages that are involved for apoptosis. So where a signal would be received, which is transduced through the cell and results in organelle breakdown and blebbing, and then apoptotic bodies are produced as a response. We'll get a little bit more detail on those. So this is a really nice summary of how you can talk about that signal transduction model for apoptosis. So one, we're talking about reception. A cell receives a pro-apoptotic signal. This can be an internal or an external signal. So that's the last part of apoptosis that I need to talk about, is what's happening in comparison with both of those. Transduction, you can simply refer to a signal transduction cascade is initiated within the cell, with ultimately results, which ultimately results in the activation of effector proteins such as caspases. So if you're going to remember any enzyme name from this process, you want it to be those caspases. What do caspases do? Take it straight from the study design. Caspases are proteins that cleave, enzymes that cleave specific proteins. So they are working inside the cell to break down those proteins that are doing different jobs around the cell, and that's going to ultimately lead to the cell collapsing. Response, the caspase cleave the proteins, the nucleus degrades the cell's DNA, the cell shrinks, and blebs form apoptotic bodies. I've seen before where you've been asked to summarize apoptosis in a 3-mark question. They are the 3 marks that question is looking for, so that you can identify what the signal is, whether it's inside or outside. Again, I'll talk about that more in a second. That you can discuss the action of caspases as transduction, and that you can talk about apoptotic bodies as a response. The apoptotic bodies being engulfed by phagocytosis, by phagocytes such as macrophages. That's one of those immune system cells that I said we're going to get to know really well soon. But you're allowed to just use the word phagocytes. That stage isn't technically considered part of apoptosis. So if you're being asked to summarize apoptosis as a process, it's everything up until this point. Here's my son with the very important questions of life.
What food is it?
Ask it again.
What food is it?
He did not get dessert. I'll have to go and deal with that in a minute. Okay, so where was I? Good. Right. Probably the most important part that we clarify for apoptosis is how we're distinguishing between the inside and the outside signals. So now that we have an appreciation of what the process is and what it looks like, these are the 2 different versions of it that you need to be able to discuss. All right, so 2 different ways that apoptosis can be triggered. 1, Cell intrinsic pathway. So the intrinsic pathway is where the cell has either been pre-programmed itself, it's written into the genetics of the cell, or the cell has suffered DNA damage. So the best example that you can use, the most reliable example that you can talk about, and what is asked about most often, is that the cell has undergone DNA damage. Once this DNA damage has occurred, then this pathway is set into motion. So you can see here the signal is outlined as DNA damage. DNA damage then triggers a cascade of events, which involves signaling from the mitochondria. So this pathway is also called the mitochondrial pathway, which activates caspases. Caspases cleave specific proteins, and the rest of apoptosis happens. So nucleus degrades DNA, and apoptotic bodies form. So that is the intrinsic pathway. Start it, if you're being asked to describe how the intrinsic pathway would occur, you start it with DNA damage, and signaling from the mitochondria is an important part. The activity of caspases, which then triggers blebbing and apoptotic bodies to form. Why is it so important that cells undergo apoptosis after DNA damage? Because the DNA is just so important. So we know that DNA is necessary to create those proteins and enzymes that help the body to grow and function. And there's every chance that if the DNA is damaged, the normal growth of that cell could be compromised, which in worst case scenario could lead to cancerous formation. So if we've got damaged DNA, definitely the cell should be triggered to undergo apoptosis. All right, in comparison to the cell extrinsic pathway. So this is where the signal is being received from outside of the cell. It is, the signaling molecule here is usually a cytokine. So if you're talking about the signal part of this model, refer to the signaling molecule as a cytokine, and it binds to something called a death receptor. And this is very much like all the other hydrophilic signaling examples we've been through. The binding of this molecule, this signal, to the death receptor will activate the internal secondary messengers, which will in turn activate, again, our caspase proteins. And what do caspases do? Get into the habit of saying it, cleave specific proteins. And then the story is very similar. So the caspases trigger that blebbing and formation of apoptotic bodies. So it's really only the signal that you need to be able to identify whether it's coming from outside the cell or inside the cell. Transduction is slightly different. If it's an intrinsic pathway, you have to mention the mitochondria, whereas if it's the extrinsic pathway, in your signaling you would have mentioned the death receptor, but both will involve the activity of caspases. And thanks to the study design, we know that we don't need to recall the order or activation of our other cytoplasmic enzymes involved here, although BCL2 will come up in a minute when I'm talking about malfunctions of apoptosis. BCL2 is worth mentioning that there's two kinds of this protein. Some of them activate and pro-apoptosis, so they will help the process along. Some of them actually act as inhibitors. So that class of enzyme, although it's not mentioned in the study design, is not a bad one to know where it's coming into play. Just back onto the extrinsic pathway quickly. So we talked about an example of how the intrinsic pathway might be triggered through the activity of UV radiation. The extrinsic pathway, a good example to use is one that we haven't actually covered yet, because it's more an example that is in the immune system, but we're coming up to that really quickly. So um, certain cells in the immune system that function in the immune system, their job is to seek out cells that have been identified as being infected. And what they will do is they will release cytokines to trigger these cells, this infected cell, to undergo apoptosis. The last thing that we want is this cell that's been infected with, say, a virus. We don't want that cell to rupture and throw those little viral particles all over the body. So the best case scenario, the best thing that can happen is that it's identified by this immune system cell, which is a T cell, and can be triggered to undergo apoptosis and be gotten rid of in a nice, neat, orderly fashion. So that would be a good example for the extrinsic pathway. Okay, and then very quickly, the last part of 6.3 is about malfunctions of apoptosis. So when apoptosis doesn't occur as it should, and what potential effects can that have on the body. So if you can have an example for both of the first two sort of ready to use, I'd imagine, and I have seen before, that exam questions related to these are pretty specific. So it'll give you an example of this and ask you how to explain how it may have come about, and you would need to identify that it was a failure of apoptosis, or ask you specifically for an example. So excessive apoptosis. Excessive apoptosis, as the name kind of suggests, is where we've got over pre-programmed kill. So where apoptosis is happening too much. And that is a really good example is Alzheimer's disease. So in Alzheimer's disease, a protein starts building up in cells, and that protein itself signals for programmed cell death. So it's where this buildup is happening in neurons, and obviously having lots of neurons start to die in a programmed fashion is not going to be good for an individual. So excessive apoptosis, a good example would be Alzheimer's disease. Inhibited apoptosis, so where apoptosis should have occurred but didn't. And the example that we've got just here in the picture is something called syndactyly. So where I referred to at the start, the webbing that is present in between your fingers as a developing embryo. That skin, that thin layer of skin in between the fingers, should be programmed to die off during development. Syndactyly is a condition where the digits are fused because those skin cells didn't undergo apoptosis when they should have. So inhibited apoptosis, syndactyly is a good go-to example. And then the third one that's specifically mentioned in the study design is cancer. So first of all, we have to appreciate that cancer it is a huge term and happens, occurs for all sorts of reasons. Two massive causes, big umbrella causes for apoptosis in very general terms, cancer in very general terms. It's either because a cell is increased in its cell division, so it's growing at an abnormal rate, or a cell hasn't died off when it should have. And so that would be a form of inhibited apoptosis. And it doesn't have to be an or. In fact, most cancers are an and. So it hasn't died off when it should have, and there's increased cell division. So that's where I was talking about the UV damage. It's important that those damaged cells undergo apoptosis because the DNA that's been damaged could be related to the growth of that cell. And so if you've now got a damaged cell that hasn't died when it should have and is growing at an abnormal rate, cancer. So it can be caused by interception at multiple points in the pathway. So if we're asked to identify a place, a point in the apoptotic pathway where this may have failed, take your pick. So go back to our little diagram here. And if we're talking about a cell that's undergone DNA damage, so we're going to be looking at the cell intrinsic pathway, any number of failures could have occurred. If you have a look at any of these enzymes, so any of the cytoplasmic proteins that we don't need to remember the names of, but either of those could have failed. So they're not formed properly or they have been inactivated for some reason. The caspases, so caspases is something that we do need to recall the name of. And just talking in general terms, we know they're an enzyme, so if they're being inhibited somehow, then that caspase can't perform its action. And because one action relies on another in this cascade, if one point fails, then the rest of it won't work. And so I think that is the end for us for 6.3. And you've got time then this week to have a look at the style activities and finish up our chapter 6 content. So getting your chapter 6 questions and your chapter 6 summary put to bed. If you have any questions, as always, please send me a message and good luck.